Hello everyone, my name is Gary O'Mealy, and in this video we are going to discuss the construction and destruction of macromolecules. In a previous video we discussed the four major types of macromolecules, that being carbohydrates, lipids, proteins, and nucleic acids, and we are going to have videos dedicated to each one of these macromolecules in the future, but for the sake of this video, we're going to be talking about how these molecules are made. And what we're going to see is that each type, generally speaking, is constructed and deconstructed in a very similar manner. What's going to make each type of macromolecule very different from one another, however, is the type of building block that is used to make them, and more specifically, what type of functional groups those building blocks tend to contain. So most macromolecules are built into what we call polymers. A polymer is a long chain of repeating subunits, and those subunits are called monomers. A monomer is a much smaller molecule compared to the macromolecule as a whole, and the idea is that we're going to take a large number of monomer molecules and form covalent bonds between them to make a very large and very long macromolecule molecule. So here, to give you the idea of how this works, we have a bunch of individual monomer molecules, and after we have finished our polymerization, you can see that all of the monomers are attached together by covalent bonds, with each yellow line representing a new covalent bond that we have forged. So forging these covalent bonds requires a type of chemical reaction called dehydration synthesis, and sometimes this is also called condensation. So if two monomers are going to be attached together, they have to have functional groups that are compatible with one another. Specifically, we have to be able to line these functional groups up and see that there is an element of water. Within the bounds of this yellow box here, you can actually see the water molecule that is going to be lost as we form this covalent bond. You can see the two hydrogens and the oxygen. So after we have formed this covalent bond, a molecule of water is lost from the reaction, and this is why this is called a dehydration synthesis reaction. We synthesize a covalent bond while losing a molecule of water. This type of chemical reaction, of course, is anabolic in nature because we are taking two smaller, more simple molecules and attaching them together to form a larger, more complex molecule, and as such, dehydration synthesis reactions require an input of energy. On the other hand, we can do the exact opposite. We can take two monomers that are joined together by a covalent bond, and we can break that covalent bond by a type of chemical reaction called hydrolysis. In this regard, a molecule of water is infused into the bond, and this is used to separate the two monomers, break the covalent bond, and reconstitute the two separate monomers that can now function independently of one another. This kind of reaction, of course, is catabolic in nature, and the chemical energy that was stored in the covalent bond that has now been broken will be released into the system, most of which, of course, will be lost as heat energy. So in theory, there really should not be a limit to how long a polymer can be, provided that we have enough monomers on hand. Here you can see 4, possibly more off-screen, monomers that are lined up together, and after repeated rounds of dehydration synthesis, we can form covalent bonds between them, and we can generally, in theory, make this polymer just about as long as we want. But generally, the polymerization occurs only in one direction, and you can see that indicated with this yellow arrow here. In particular, we're going to see that's the case with protein synthesis and nucleic acid synthesis, in the idea that polymerization only occurs in one direction and not the other. However, there is one thing that we are going to need in order to get efficient polymerization, efficient construction of macromolecules to occur, other than just the monomers, and that is the presence of an enzyme. In a previous video, we discussed how chemical reactions that happen inside of the cell are very slow unless you have a chemical catalyst present. And since enzymes are the catalysts that we find inside of cells, dehydration synthesis and hydrolysis reactions are both going to require the presence of enzymes to catalyze those reactions. In this table here, you can kind of get the general idea of what kinds of enzymes catalyze the synthesis of macromolecules and the hydrolysis of macromolecules. So for carbohydrates, the type of monomer is called a monosaccharide, and I have under synthesis and hydrolytic enzymes, I have it being varied. That doesn't mean that there's different types of enzymes that do it. It means that it varies in terms of what we call them. In particular, one that you may have heard of, an enzyme called amylase, is a type of enzyme that breaks down the glycosidic bonds that join monosaccharides together in carbohydrates. In proteins, the monomers are amino acids. The ribosome is the molecular machine that has the enzymatic activity that builds the bonds between amino acids when building proteins. And then in a previous video, we talked about proteases like trypsin and pepsin that break the bonds between amino acids and digest proteins. Nucleic acids use monomers called nucleotides, and these are built by enzymes in the cell called DNA polymerase and RNA polymerase. And enzymes called nucleases break the bonds between adjoining nucleotides and digest DNA and RNA molecules into their monomer components. And then you're going to learn in one of the next videos when we talk about lipids that there are all different types of lipids, and there's a little bit of variation in terms of how they're actually built and broken apart. But generally, the type of lipid that follows this type of construction and deconstruction paradigm that we've talked about are triglycerides. They are built by attaching glycerol to a number of fatty acids. The synthesizing enzymes tend to vary in terms of what they're called, but they are broken down by a general class of enzymes called lipases. Okay, that's going to do it for this video. Here is a list of vocabulary terms you probably ought to be aware of after watching this video. And for checking your understanding, number 1, which of the 4 types of macromolecules is there a definite limit to how many dehydration synthesis reactions can be used to make a polymer? We talked a little bit about that in the table that we just looked at. And then number 2, how could a cell's control over the activity of its enzymes determine the stability of the cell's macromolecules? Okay, that's going to do it for this video. Our next 4 videos are going to tackle each of the types of macromolecules one by one. Hopefully you'll look forward to it. If you have any questions about this video, go ahead and drop them in the comments section, and I will see you next time.